if we will go for details of a particular DNA which is available to us in a very small quantity, or if we will go for its further use, we need to multiply or amplify its number. To get pure, identical and multiple copies of that particular DNA, we are using certain technique like PCR and gene cloning. Hello everyone, I am Suchi. Welcome to my channel. Let's discuss today about PCR. PCR, or you can say polymerase chain reaction. In this chain reaction, one copies of DNA form multiple copies of similar type by passing through a chain of reaction. This chain of reaction carried out in vitro. In vitro means it doesn't require any biological body to carry out this reaction, or you can say process, to multiply DNA number. In vitro means lab, which is outside of the living body. This chain reaction first invented by Mullis of Citrus Corporation of California in the year of 1985. He got Nobel Prize for this invention. In this chain reaction, we are getting multiple copies of desired DNA. To carry out this process, first we need a sample DNA which we want to multiply its number. Normally, DNA present double-stranded helical. And both the strands bind with each other by hydrogen bond. These are the hydrogen bond. So this will act in this reaction as a template. Second, we need two type of oligonucleotide primer. This primer contains 10 to 20 nucleotides. It will bind at the 3' end of each strand of DNA. This one is the 3' end. One primer will bind here. Another one primer will bind here at this end. And it will provide free 3' OH end. Because this one is the 5' end of this primer. This side is the 5' end of this primer. It will add one nucleotide at this free 3' OH end, which will be the complementary base of this strand, existing strand, and the polymerization process will occur from 5' end to 3' end direction. Next, we want for this reaction different type of deoxyribonucleotide. Like D-ATP, D-CTP, D-GTP and D-TTP. This adenine, cytosine, guanine and thymine, these are the base required for the DNA, because DNA having these adenine, cytosine, guanine and thymine as its nucleotide. To make new strand, we need different nucleotides. So, we are taking these nucleotides as reagent to add one nucleotide at this 3' end to make another one new strand. So, fourth one we need DNA polymerase. This DNA polymerase is an enzyme which helps to bind this nucleotide at this 3' end to make or form a new strand, which will be the complementary strand of this existing strand. This DNA polymerase enzyme is highly sensitive to heat. In high temperature, its enzymatic activities will reduce. So, for that reason, we have to add, after each cycle, we have to add this DNA polymerase enzyme. To avoid this problem or to solve this problem, many other stable enzymes isolated from various bacteria like Taq DNA polymerase, Vent polymerase, and PFU polymerase. This Taq DNA polymerase isolated from Thermus aquaticus. Vent polymerase isolated from Thermococcus littoralis. This PFU polymerase enzyme isolated from Pyrococcus furiosus. These enzymes are thermostable in nature. In high temperature, it will not lose its enzymatic properties. So, these enzymes are used in this chain reaction to carry out this process. First, this Taq DNA polymerase enzyme isolated by Lauer and his group in the year of 1989. Including this, we are also adding trisacl buffer to maintain pH, cofactor like magnesium chloride, potassium chloride to carry out this process, and mineral oil to prevent evaporation in high temperature. Normally, this process carried out in a single tube. We are keeping that single tube with reagent in thermal cylinder, and that thermal cylinder programmed in such a way that temperature will increase and decrease automatically and alternatively to carry out this process. In this process, normally 3 steps are there. One is denaturation, second one is primer annealing, or you can say primer jointing. Third one is polymerization, or you can say extension. To start this process, first temperature will rise up. DNA is double stranded. When temperature will rise and reach at 95 degree, or if we take in range between 90 to 98 degree during this range, and it will maintain for 1 minute. During this period, this hydrogen bond which is in between this double stranded DNA, it will break and form two separate single stranded DNA. In this high temperature, this hydrogen bond will break, or you can say it will melt. And this process is known as, or breaking of this double stranded DNA to form two separate single stranded DNA is known as denaturation process. After this process, temperature will fall. When it will reach at 55 degree, or if we will take in range between 40 to 60 degree, and this temperature will maintain for 1.5 minutes. During this period, one primer will join at this 3' end of one strand, and other primer will also join the 3' end of other strand, and it will provide free 3' wedge end. Because this end is 5' end of the primer, this joining of this primer at this 3' end is known as primer annealing. Once again, temperature will rise up. After this process is denaturation, and this process or joining of primer at this 3' end is known as primer annealing. Once again, temperature will rise up. When temperature will reach at this 72 degree, or if you will take in range between 70 to 78 degree, and temperature will maintain for 1 minute. During this period, 1 nucleotide will add this free 3' wedge end of primer, which will be the complementary to the existing strand. If here it will be A, here it will add T. If it contains C, that time it will add G. Likewise, A will bind, or A will join complementary to T, and C will bind complementary to G. In this way, 1 nucleotide will add and form complete new strand. This process is known as polymerization. After this process, it will form two complete DNA strand. This one will be the new one, this one will be the new one. After first cycle, second cycle, enter in similar process and form two new strand. In this way, after 30 cycle, we will get million of copies of DNA of similar type. This technique is highly efficient. It doesn't require any costly restriction enzyme, ligase enzyme and vector DNA. It doesn't require any skilled worker or biological body to carry out this process or multiply DNA number. Within very short span, we will get million or billion number of copies of DNA which will be the pure and identical. On the basis of primer we are using in this process, this PCR technique is of different type like single-sided PCR, inverse PCR, random PCR and RT-PCR. Recently we have used RT-PCR for diagnosis of this coronavirus. This PCR technique helping in various ways to solve multiple issues by amplifying DNA which we are getting the incidence place to identify criminal in forensic science. It solves parental issue like identify real parent of a child. It helps to identify genetic disease, pathogenic disease and the presence of a particular DNA or particular gene in a DNA, and it also helps to build DNA library. This is all about PCR. Thank you.